remember it. And so the computer scientist in me was wondering, well, where's the file? Where's the hard drive? And what language is immune memory written in? And so it led me down a long rabbit trail in immunology. And I got kind of curious about this idea of things that we couldn't vaccinate against. And like, take HIV, we haven't done a good job of that. And if you look at HIV, it's killed countless tens of millions of people. They all made trillions and trillions of antibodies and then eventually succumbed to this virus. But a handful of people made an antibody that kept them safe. And we call them like elite suppressors or elite controllers. And we've pulled these antibodies out of the arms of survivors in Africa. But if you think about it from kind of an information science point of view, like we basically waged a brute force attack with flesh and blood against a computer program, against a virus. And we paid a high price for it. And now we have those sequences, but I got to wonder, could we use the computational power of the planet instead of flesh and blood to fight that battle? And what is actually the theoretical key space of the humoral immune system anyway? Like how many possibilities are there? And that, especially at the time, was pretty heretical in the field of biology. Like now it's much more common, I think, to like mix parlance and logic between biology and computer science, but back then I think it got ill looks. And as I went down the path, though, some snarky scientists pointed out to me that cells don't have USB ports. And so even if you have the code, you can't upload it. And so that seemed absurd to me, and maybe more of a profession of my own ignorance of the field. But I said, well, we should fix that. We're going to build an app store for the human body. And so I started this company called Immusoft, and we were trying to make autologous long-lived plasma cells that would secrete therapeutics. And so these are the cells that specialize in making antibodies. They're responsible for the bulk of the antibodies you have circulating in your blood now. And what's really fascinating about them is when they are first created, they will either die in about a week from just the ER stress of making that much protein, or they get to these survival niches where they're effectively immortal. And so average lifespan in humans is like 20-some-odd years. It's a long, long time. In fact, if you take blood from an 80-year-old, you can still find antibodies against smallpox in it. And they're made from the same cells that were made all those years ago when they saw the virus or the vaccine. And so we're going to build this. We're going to kind of hijack this system to make arbitrary proteins. And it actually led me here to Caltech. And so we licensed the first technology for this out of David Baltimore's lab. And he had built this in vitro lymphopoiesis system that could take a CD34 cell and go all the way to a long plasma cell in culture. And that is not super practical therapeutically. It took 10 weeks. But we're able to modify it with a cool virus we found in Ines Leon and drop the time from 10 weeks to 10 days, and later get rid of the virus entirely and drop it to 7 days. And so, next slide. And so this was the first engineered B-cell therapy to ever go into human trials. And the process is similar to like what you think of with the CAR T. You take the cells out, program them, put them back. But next slide. You're gonna get tired of this. I put in maybe 2 next slides. But it did something really cool. So this went into clinic a little over a year ago. And it was a long, painful journey, which we can get into later. And I learned a lot about the regulatory system. But this is the first patient treated. And in this case, we're making an enzyme for a rare lysosomal storage disease called MPS I. And what's sweet about this is, well, this disease is lethal, right? And if you have a severe form of it, standard of care is a stem cell transplant when you're about 2. And if you have an attenuated form, it's enzyme replacement for life. 400 grand a year, every week you're getting pump full of this. We modified his immune cells to make this enzyme. And this is him at the year mark. He's actually walking as well as a healthy age-matched person. Like, we beat the standard of care like 5-fold. It's just amazing. And next slide. All these things like joint mobility, stuff that you can never reverse. It's actually pretty amazing, especially given how low the dose was. Like the FDA made it start very low, and I suppose they almost killed us by putting us on clinical hold for 2 years. But then they also said collect all the endpoints that Aldurazyme used for approval, which is very odd. Like a phase 1 trial at a low dose where you don't really expect to see much. But we're in no mood to fight, and we did it. And it came out way better because of it. So I guess a little beer in the end. And so these patients are saying, like in cross my legs for the first time. I can like touch my back in the shower for the first time. Like, it was really pretty remarkable. And so the second patient was dosed fairly recently, and actually just today they were able to redose this patient with a higher dose, which is pretty amazing. So, and that's something that's unique about this kind of therapy. A lot of gene therapies you can't redose. Next. But there's a problem with these advanced therapies, and it's one that I suppose was particularly like poignant to me coming out of computer science. And like in the world of computer science, you can basically have a handful of amphetamines and a few all-nighters and knock out a minimum viable product. Like everything's open source. Like you can write code. You can move very quickly. It's not like that in biology. And if you think about like the history of medicine, for almost all of human history, medicine is plants and prayers, right? Small molecule drugs and rituals. And it wasn't until very recently that we really started getting into biologics and then later gene therapies and gene-modified cell therapies and now genetically modified humans. But as we gain these really impressive, powerful tools, the ability to make them has become more and more challenging. And so like right now, I mean, I'm 100% confident the people in this room, if you needed a small molecule drug, could make it. You can make it. You do a lot of medicinal chemistry in your bathtub, but you cannot do that with a biologic. You're going to kill yourself. And so this bothered me a bit, that I felt like the one person who had no say in medicine was the patient. And like, you know, the doctor won't give it to you, you're screwed, insurance won't cover it, screwed. If it's not proved, just fuck that, like, you know, you're dead before it ever gets to trials. And but something interesting and unexpected happened around the same time, and that's the cost of DNA has been falling faster than Moore's law. Like right now, you can have a gene sequence into your iPhone, and it'll show up in the mail in a FedEx envelope. And so now you can write a program in the language of life, and if you can find a way to upload it to yourself, then you could make a drug for free or for cheap. So next slide. And delivery is the problem. Okay, good. So right now, like the world of particularly in vivo delivery is dominated primarily by actually two of these modalities, the LNPs, which we all are associated with, like the COVID vaccines and AAV viruses. And they both have pros and cons, and we'll kind of come to them later, but they aren't super well suited to making things in a primitive environment. So go to the next slide. But electroporation is kind of an interesting one. And this is one that even like so Emisoft's clinical program uses electroporation now. It's very old technology. Like if you basically tase a cell, you can get things inside of it. So you run an electric field across it, puts little holes in the membrane, nearby things can get inside. And DNA is a slight negative charge and can actually be drugged this way. And strangely enough, the same thing actually works in a living creature, although it becomes more challenging. And so you can basically inject DNA in the middle of a needle array and plug it into a light socket. And as you can see from the potato there, you can cause some damage if you do it incorrectly. But next slide. But if you do it right, you get expression of a gene of interest in your tissue. Assuming you've encoded the DNA correctly, it will make foreign proteins. And muscle in particular is actually pretty good at making protein. Next slide. The problem is, it hurts a lot. And before I went things too insane, so there's a story about this. The first time I was working on this project, I was trying to build an open source in vivo electroporator, by the way. And I wanted to use this to make Ebola antibodies in Liberia. And so I had the CEO of MapBio to give me the gene sequences for them. And we modified them from mammalian expression and put them in some monkeys at UW. But no one had ever done this on a monkey. I couldn't find any published data for it. And this device I initially had cobbled together from parts on eBay, I wasn't really sure what it would do. And I was reluctant to do it, so I zapped myself with the device. And I promised them I wouldn't put any DNA into it without their consent. But I can attest that it does hurt. Next slide. And you can buy these things commercially, but due to them hurting and having other issues with not being able to modify tissue like muscle reliably, I was trying to make something that you could use in a more primitive environment. And so, next slide. So this is like an early version of this open source electroporator. So this is an old ECM device I made off the BTX from eBay, and I kind of custom built this needle array to go around it. We made it so you could put DNA into the middle. And next up. And so in this study, though, we made a bunch of DNA in our lab. It looked like Breaking Bad. It's a bunch of giga preps everywhere, just gobs and gobs of DNA. And we put them into these monkeys. And for a few $100, we made a treatment that probably would cost over 100 grand. You go to the next slide. And so this is the data from it. So in its high dose, it seemed to be high enough for post-exposure prophylaxis. And I mean, that's pretty remarkable, right? Like you can make DNA very fast, and you can make it with almost nothing. It's a vat of Ebola. And so this kind of really was the first example of a tool like this that I thought, well, you could actually begin to kind of shift the balance of power. And in the meantime, something really strange happened. So like I said, I had a hard time getting our first treatment into clinic. This is the first therapy of its kind. It got a lot of scrutiny. And that's been true of gene therapies in general. They're among the most regulated things in medicine. But a few years ago, something really strange happened, and we gave hundreds of millions of people a gene therapy for an infection they didn't have. And I'm not ignore the RNA stuff for a moment, because I think it probably should have been regulated differently. But the adenovirus-based COVID vaccines are quite literally a textbook gene therapy. It's a DNA virus. Like, it's not a controversial statement, although it might be a charged one. And AstraZeneca, J&J, CanSino, Sputnik, they're all adenovirus vectors. And these are things, in all fairness, have killed people before, like at albeit much higher doses, but they weren't a gray area anymore. And so from a policy point of view, though, I think this is actually amazing. It may prove to be the one thing that makes the longevity field possible. Because I don't really believe that we're going to be able to cure aging with pills. And I mean, we've boiled the ocean in medicinal chemistry. It's not for lack of trying, and we still could go into this. But I think that we're going to need to rewrite the code of life if we want to really meaningfully impact human lifespan and health span. And so before COVID, there was no way to take any gene therapy into anything other than really nasty, lethal disease when there weren't other good options. And now, from a policy point of view, that door is basically blown off its hinges. And I, for the record, don't want to be making this argument to FDA, but I do think that policy will matter, and I think it's incredibly relevant to this field. Because if you can take a gene therapy for a disease you don't have, surely I can take one for something I do have. And we'll come back to this later, because it's going to become very real very fast. Next slide. So the journey through, the journey through delivery, the electroporation is a crude tool for in vivo use, and making things like viruses at home is a very bad idea. And so we really need better tools, and that's not just for doing things in the developing world of the garage. We need better delivery of, let's say, heal in genetic medicine. It really is pervasive in space. So let me talk a bit about some projects we've been working on, and this one I'll only touch on briefly, but it is an interesting story of how disciplines can mix. So in that time when I was trying to make my open source electroporator, I eventually got in over my head with electrical engineering, and I was trying to make it solar powered at the time, which was a challenge. And so I got connected to a brilliant electrical engineer who's actually the CTO at Helion, the fusion power company, the pulse power expert. And I asked him for help with this, and he said, why don't you use a magnetic field instead of an electric field? And so the downside, of course, as we saw from the potato, if you run too much power through it, you get myocyte chip cookies, basically a burn mark and no expression. And the magnetic field wouldn't have that problem, right? There's no arcing, there's no point-to-point flow. It's an induced field. And so, next slide. And so it's a general idea. So similar concept to electroporation, but there's no electrodes anymore. It's a field. You can think of it a little bit like going through an MRI at Mach 2. So it's like a soul-crushing multi-Tesla field in a thousandth of a second, just like boom. And you get that little burst of induced current and then nothing. And so I can't keep running. And so actually, the first experiment for this, he had cobbled together a bunch of parts, pilfered from somewhere, and brought them over to my lab one night. And so I naturally just draw some blood and put the Epinorth tube and stick it in the device and put some GFP DNA in there. And no one really knows how many times you should push the button or what the setting should be. It was roughly designed to mimic field conditions of electroporation, but who knows? And so we hit this a few times, took out the cells, threw them in the TC hood, and said, oh, the scientists isolate them and look at them in the morning and see if there's GFP. And lo and behold, we got transfection of the PBMCs in whole blood, which is something you can't do with electroporation. And that was really cool. But we didn't have the bandwidth to really start a company on it, so we started the company, but then we found a CEO and we shipped off to Y Combinator. So it turns out that years later, one of the primary things it's up to is trying to load things into red blood cells directly. Something we never looked at the time. But the Department of Defense found out that if you put like Trailose in a red blood cell, you can lyophilize it. And the idea of making powdered blood that's shelf-stable is very valuable. So I've been doing that. Next slide. So this is, we had till 5:30. I want to leave time to talk because it'd be more interesting than listening to me. So this is what I'm up to now, though, and kind of where I've landed on delivery lately. And so we're out building these genetic medicines, and we use a really cool delivery technology called a PLE. So next slide. And like I said, the kind of dominant players for in vivo delivery are these LNPs and viruses. And the problem with viruses, I suppose, is although they're purpose-built machines for nucleic acid delivery and they're good at it, your body knows what a virus is and it doesn't like it. I mean, life's been waging war against viruses since life existed, and there's far more viruses than living things out there, so they're very adept at trying to thwart them, and they get very unhappy when you put lots of them in. And the LNPs are easy to make, relatively cheap, but they typically cannot deliver DNA because they become wildly toxic, and they have a profound tropism for the liver, and so pretty much no matter where you inject them, they end up in the liver. I'll show you more about that later, but you can go to the next slide. So our goal here was to kind of make the best of both worlds. So something that had the manufacturing attributes of an LNP, but the entry characteristics of a virus. And we published this in Cell just at the end of last year. A great paper, highly recommend it. But this little fast protein here is actually how it gets in. And actually, we can go to the next slide. And the issue with LNPs is they kind of made a Faustian bargain of sorts with lipid chemistry. And what makes them possible are ionizable lipids, so lipids that will change their charge when the pH changes. And this is critical because the LNP is endocytosed by a cell, and it has to get out of the endosome. And so as the endosome's pH changes, they develop this charge, and they blow a hole in the endosome and escape. Poorly, admittedly. But that's what makes them work. If you take that away, they're not toxic anymore, but they also don't work anymore. This, though, uses, I don't know, is any virologists in the room? Yeah, this is nerdy virologists like this. So this uses a little fusogen from a reptilian fusogenic orthoreovirus, and initially one that infected the stomachs of alligators. But it's archaic for scale, and it's next to the HA protein from influenza. And this is the smallest known fusogen that's capable of membrane fusion by a factor of 2 orders of magnitude, so it's 100-fold smaller. And it's from the only non-enveloped virus even known to have a fusion protein. So it's like really, really wildly different from like an evolutionary point of view. And the virus actually doesn't use it for entry, it uses it to build syncytia, to fuse adjacent cells together. But a crafty virologist in University of Dalhousie got curious about how these viruses worked and tried to like isolate and characterize this mechanism. And the first thing that slowed him down was that it kept running off the edge of his gel because it is so much smaller than what he expected to find. And because of that size, it is also not very immunogenic. I mean, computationally, it doesn't really have much in the way of B-cell epitopes, but it's only about 12 amino acids long actively. It sits there in the membrane. And so you can administer this lots and lots of times to an animal or a person, and they don't get anybody's against it. And so this, what it does mechanistically is it will fuse directly with the membrane of the cell it comes in contact with. And so there's no receptor interactions, no endocytosis. And so next slide. And so this lets you do something really wild. Like I said, the LNPs made their Faustian bargain of sorts, and like half of them are, half of the contents of LNP are basically ionizable lipids, this thing that is toxic but essential. Then third cholesterol, and then they can mess with the other parts. And I don't know if you hang out at delivery conferences ever, but every year there's hundreds of posters about how they're
changing the tropism of LNPs, and they've made very little progress. It's because they really can't change that much. But if you don't have that in the equation anymore, then the parameter space you can explore in lipid chemistry is huge. There's countless thousands of lipids with all sorts of different behaviors that you can incorporate into a particle like this, and since it's using the fusion protein for entry. And when I got started with this, we were trying to make a senolytic. And this is after a paper that's kind of famous in the space. It kind of launched the field, the Baker paper. And these guys had made a transgenic mouse that put an inducible caspase under P16. And so they could wipe out all those cells with an otherwise innocuous drug. And it was the first paper, I think, to really show that doing that extended lifespan and health span meaningfully, like most pathologies of age were attenuated, they lived 20% longer, 25% longer. And but in the grand tradition of academic science, it was translationally useless. You can't go back in time and modify your own embryo after all. So like, how would you do something like that? And so when Ocean was started, we wanted to make something that was clinically viable. And to do that, you'd have to deliver nucleic acids across the whole body, a massive dose and great penetration. And so when I started looking for a delivery tool, I said, I want something that has no tropism, a flat biodistribution curve, and get every cell in the body. And which is kind of a laughable request if you're in delivery. But I think this might be the closest anyone's going to be coming for a while. And so this is IV in a monkey, 1 mg per kg. So dose that would be lethal with DNA and LNP for sure, but it's fine in here. You'll notice the first thing probably is that the liver is no longer at the top of the list. This one, the lung is, and that I think is mostly just due to biophysics. Like it fuses with any cell indiscriminately, but it needs to be in contact with the cell. And so there's lots of places for it to meet cells in the lung. And we have versions of it now that can do wildly different things. But this is kind of where we started. And the goal was that I wanted to take targeting out of the realm of chemistry and into the realm of information. So I wanted to target with logic gates in DNA and have delivery be ubiquitous. So next slide. And this is an illustration of exactly what I was up to. So on the left there's a mouse that got IV administration of DNA encoding luciferase under CMV promoter, and as you'd expect from that last slide, it goes everywhere, all the tissues light up. But here on the right, that mouse got the exact same dose and had the exact same route of administration. The biodistribution is unchanged, but now it's under control of a synthetic, like engineered version of the P53 promoter, tumor suppressor. And there's a tumor implanted in here in the red dashed line, and you can see that the expression is entirely restricted to the tumor. So we're basically, the idea is that a cancer cell knows it's cancerous. It's transcriptionally different enough that you can target it based on that. And so this is what we're doing with senescent cells, but senescent cells are all in the body and not a nice little lumped image. Okay, next slide. Okay, so this is what we're actually doing. In that video, there's time-lapse microscopy of an adipocyte dying, which is actually for a much newer program. But we were expressing inducible caspase also. And so you could kill like one target cell here and not hurt any of the cells next to it. And this is really important in aging because, humans have been good at killing cells for a while now, but usually they cause a lot of collateral damage. Like nobody thinks chemotherapy is really pro-longevity. And so this doesn't have a drug, it has no poison. Like it's just a computer program written in DNA. And I think that this opens the door to do all sorts of really fun stuff you couldn't have dreamed of before. This is an example of how much you can tailor the PLV to hit different tissues. And so broadly speaking, we have, although I was obsessed with this one domain of targeting with information, there's kind of 3 that we can use. One would be that, the other is the lipid composition themselves, which can confer really profound tropism on their own. And then we can also add surface targeting to it if we want. But here you have, this is a CNS targeted one that was done with Entos and Lilly, an ocular one. I can't see my killer examples. Talk trash about LNPs more. If you inject like an LNP into your eyeball, it's in your liver in hours. This is actually why the COVID vaccines work so well, right? They express in the liver, it's like massive protein factory making gobs and gobs. It's like, and we made a COVID vaccine instantly with this vector, and it was very challenging to rival those antibody titers because this doesn't actually traffic that way. And here, like you go up to the lung and you don't touch the liver at all. This is a Cree-based assay, so it's incredibly sensitive. And you really need to hit the cell once and it will light up. And near and dear to my heart is muscle formulation, which is a topic of interest now. But you can do this. There's now like hundreds of these formulations that do all sorts of fun stuff. And next slide. And this is, I'll talk a little bit about what we've done so far, and then I will stop talking. But these are the programs that we're messing with. So go to the next slide. So this, we talked about a little bit before, this analytic, and just an example of it killing cells in a co-culture. This is like frame grabs from the time-lapse microscopy, and both examples are due to P16 and P53 like this. Next slide. And then probably the most advanced program we have is actually this frailty. And the idea is that there's a few things more central to aging than frailty. Like, it's not just that, loss of muscle makes you less resilient against disease and injury. It affects your cognitive function, your metabolism, your social interactions, quality of life, all this stuff. Unfortunately for us, the pathway that regulates skeletal muscle is actually really well understood. It's studied for a long time. And in fact, that cow there is a naturally occurring gene mutant. It's deficient in a gene called myostatin, a negative regulator of muscle growth. And the classical kind of phenotype for that is you have about twice the muscle mass of your wild-type equivalent. Follistatin overexpressor as a transgenic is actually twice the size of that. And those don't occur in nature, to my knowledge, but you can see that mouse. It does not look like the mouse you want to be handling at the vivarium. It's a monster. And this doesn't, unlike say like an anabolic steroid or something that's like a master regulator, all sorts of stuff. This is the same between men and women. Like it's not a androgenic hormone at all. It just regulates muscle growth, and it does so in response to exercise typically. And so what we're effectively doing is we're tricking the body into thinking it's working out when they're not, and so it just gets bigger. And this has been something that pharma has attempted to turn into a drug for, gosh, over a decade. And it's actually quite difficult to make it recombinantly. It requires some post-translational modifications that are difficult. And doing it with like a viral-based gene therapy, although it's been done in humans several times, is cost prohibitive and probably too dangerous to apply to just an old person. So go to the next slide. This is actually in the Cell paper. We give the mice a single administration of this, IV, and in 6 months, they're 50% larger and twice as strong. And at some point, you're going to see a whole bunch of scientists who are really jacked, and it will have nothing to do with any of this research. But it's really quite strange, quite profound. And even if you do it in a very old animal, they don't get as big, but they actually still get meaningfully stronger. And so the goal for this, like, so I don't want to make something that costs $1 million that a few people can have. I want to make a treatment that's cheap enough that your healthcare payer is going to pressure you to take it because it will be cheaper than frailty. And I think that should be like the objective for any of these longevity therapies. That I think it's perfectly fine to use billionaires as a model organism, but if you're building them out, we want these to actually be broadly deployed. And that thing, so the cost of DNA is falling fast, and about 80% of the cost of goods is DNA. And if it works as expected in humans, it would last for about a decade. So it'd be long-lived, but non-integrating. It's durable autosomal vector. This is a program I'm working on now, and it's been, consuming all my life for the last couple of months. So AbbVie Ventures led our Series A, and they got very interested in applying this technology we're using in oncology and analytics to fat. And the goal would be to get effectively Botox for fat, to be able to do body sculpting with a hypodermic needle. So we're not talking about weight loss anymore, targeted fat removal. And we're looking at this for a rare disease initially, lipedema. And that's a disease that is kind of characterized by abnormal fat deposition, mostly in the lower extremities. It's quite debilitating, but go ahead. And so for this study, I'm going to show you in a minute, we did this in pigs. And so you just inject this into the fat of the skin, and we've mixed this thing with GFP and the suicide gene so that we can see what cells we transfect and activate them in some. And in theory, our little readouts will go like that. We can go next slide. And this is just a histology sample from this latest study. And so you're looking at distance from the injection site going out. And you can see the adipocytes here, and they're not adipocytes over there. And I think it will spread beyond the system after a week, and so I won't read a huge amount into it. It was taken more for tolerability than anything else. But this idea that you could selectively ablate tissue without any invasive procedure, I think would be very lucrative. But more importantly, actually, go next slide. So, get it myself. I think there. More importantly, I think for the space, and if this gets approved, it will be the first ever approved cosmetic gene therapy. And think about that. Like, so back to this original point about gene therapy sort of infections you don't have. If you went to FDA 5 years ago and said, "Hey, I'd like a cosmetic gene therapy," that you would have a very short interaction with FDA. It would be over before it started. Under no circumstances would that have flown. But now we have this precedent, and you've got a big pharma that's serious about taking forward for that. Like they asked them. Well, we had our first regulatory interaction for lymphedema, and like now we're going cosmetic, and so in a period of 5 years from now, you might be able to have a cosmetic gene therapy, and that I think is absolutely essential for the longevity field because it will now have made gene therapies elective, and if you can do that for a cosmetic reason, you can do it for all sorts of mechanistic aging things that can't be tied directly to a disease. Like one of the reasons we didn't pursue this analytic wasn't because it didn't work. It works great, but the regulatory path is really rough. Like everybody generally agrees senescent cells cause trouble and they accumulate, but tying it to something you can get through a phase 3 endpoint is a lot different beast. And so if you take that away, then all these therapies could potentially go to market under that designation. So I think it's huge. This slide is just basically kind of a non-con, human-readable version of our field of use for the technology and what we're doing. And so when we put the company together about a decade ago, so we're the first group to use it for nucleic acid delivery, and we placed a big bet on the technology early on. And when we had our choice of which any field we wanted. And so what we did was we selected some things that we thought, that FDA would approve, a big pharma would license, where we'd start. But we also got rights to a bunch of things that weren't actually possible yet. And the thesis was that delivery is the Achilles heel in genetic medicine, and as science progresses, we'll begin to elucidate these pathways of aging that we don't understand today. But as we do that, we're going to need to intervene somehow, and delivery will become even more essential. And so we wanted to basically build an empire for this over a long period of time, not build a simple asset that's only going to flip. And so I'll leave it there because it's a little longer than I wanted. But I think that, as odd as it sounds, what we really need is a big pharma for longevity. And the reason I say that is even if you look at companies like Altos, $3 billion Series A, or like largest Series A in history, it's nothing compared to the R&D budget of a $100 billion company. And I think that if we want to meaningfully tackle something as monumental as aging, that's less. Tackle something as monumental as aging, we're going to need the resources of a big pharma, and we shouldn't shy away from doing that. Like that, we can build these things out, we can license them early on, but what we really want going forward is the ability to take these all the way through to commercial.